Assalamualaikum and hi everyone. Today we will talk about a novel herbal formulation, Glymin sprinkle, enriched with multigrain, inhibits oxidative stress, AGE formation, and pancreatic pathology associated with diabetes. My name is Nasnurul Fadhilah binti Nasir 049985. These are our table of contents. First one is background. Diabetes is a multifactorial, heterogeneous metabolic disorder with micro and macrovascular compilation, which means that treatment becomes more challenging due to adverse effect of current medicines. The use of dietary food supplement is the most advocated, mainly derived from medicinal plants, fruits, vegetables, and grain. The objectives of this research is aim to investigate the effect of novel herbal formulation, Glymin sprinkle, enriched with multigrain, inhibits oxidative stress, AGE formation, and pancreatic pathology associated with diabetes. The third is the introduction. Diabetes mellitus is a chronic metabolic disease which causes hyperglycemia due to insulin deficiency or resistance and interrelation between the genetic and environmental factors in contributing beta cell dysfunction. The factors for diabetes mellitus are lifestyle changes and unhealthy food pattern. This factor will cause upsurge in diabetes. The exact mode of pathogenesis of diabetes is still unknown. Oxidative stress plays a crucial role in the pathogenesis of diabetes. The reactive oxygen species ROS generated during a stress condition will scavenge by antioxidant present in our body. Diabetic condition may cause excessive production of free radicals. Antioxidant activity will be diminished considerably. This can lead to the severe damage of macromolecules and the formation of advanced glycation end product AGE. AGE is a complex heterogeneous compound formed via non-enzymatic glycation of biomolecules. This will trigger the activation of intracellular pathways. AGE is one of the major contributing factors, leading to insulin resistance and causing damage to the cells. This will cause development of nephropathy, retinopathy, neuropathy, and coronary artery problems. The treatment of diabetes. There are many treatments of diabetes nowadays. The first one is oral hypoglycemic drugs, which are glibenclamide, metformin, acarbose, and insulin injection. These anti-diabetic drugs are prescribed long-term to lower the blood glucose level, but prolonged usage will lead to organ damage. The second one is traditional herbal medicine, the preference of people as their primary healthcare remedy. Natural products effectively capable of managing diabetes. Polyherbal formulations are very efficacious, which can control the diabetes and diabetic associated changes in the body. Recently, jackfruit, Artocarpus heterophyllus, is known for their medicinal value. They possess different pharmacological activities, for example, anti-diabetic, anti-inflammatory, antioxidant, and antimicrobial activities. The fourth one is methods. Methods used for this research is, the first one is report the effect of novel herbal formulation, Glymin sprinkle, enriched with multigrain on streptozotocin-induced diabetic model. The second one is diabetes was induced by a single dose of streptozotocin, 60 mg per kg body weight injection through intraperitoneum. The efficacy of Glymin sprinkle will be analyzing various parameters like antioxidant status, liver enzyme markers, glycation of hemoglobin, blood glucose level, glycosyllase 1 protein expression, and histological analysis.
My name is Alia Amira 049980. So I will continue with the results. For effect of GS on blood glucose level, the results show that the glucose level increased in the diabetic control group in each respective day as compared to normal. But GS treatment gradually decrease the glucose level and restore to the normal level better than the standard drug over the period of 21 days. For effect of GS in glycated hemoglobin level, in diabetic control group, glycation of hemoglobin or HbA1c was significantly higher as compared to the normal group. But for effect of GS on pancreatic antioxidant enzyme, streptozotocin induction results in the amplified production of free radicals. Thus, the antioxidant levels was significantly reduced in the diabetic control group. Next, effect of GS on GSH level. Comparing the diabetic control group with normal, the non-enzymatic antioxidant GSH was reduced significantly, and the effect of GS on liver enzyme. The major marker enzyme SGOT, SGPT, ALP were significantly increased in streptozotocin-induced control group compared to normal, which clearly shows the liver damage. Next, for effect of GS on lipid peroxidation end product. As a result of lipid peroxidation, TBARS level was elevated significantly, which implies the overprotection of free radicals. After that, the effect of GS on the level of glyoxylase-1. The result shows that the expression of glyoxylase-1 protein is considerably suppressed in the diabetic control group as compared to normal, and it was significantly expressed in GS-treated rats. And the last one is histopathological analysis of pancreas. The pancreas contains lobules of pyramidal cells with zymogen granules arranged in EX unit and also normal population of islet of larger hen. Okay, I will proceed with the discussion. Streptozotocin-induced diabetic model is commonly used. Streptozotocin will enter into the pancreatic beta cells through glucose transporter 2, which cause DNA alkylation and trigger the activation of poly-ADP ribosylation. So NAD plus and ATP will be depleted, and oxidative stress condition will be created that leads to the formation of free radicals, results in the pancreatic beta cells necrosis. When level of HbA1c increase, it shows signs of hyperglycemia. So from this study, diabetic patients tend to have higher level of glucose, so that it leads to the glycation of hemoglobin and thereby increase the level of HbA1c. As a result of hyperglycemia, the normal functioning of pancreatic beta cells will be distressed, that lead to the stress condition due to the formation of more and more ROS and also RNS. So GS treatment significantly reduce blood glucose, which means it helps to improve the glucose metabolism and inhibits the glycation of hemoglobin. Next, SOD, CAT, GPX are the major scavengers of radical which act as a first-line defense against them. GSH plays significant roles in the protection of cells from oxidative stress. So in this study, increase the level of SOD, CAT, and also GPX and GSH. As a result, reduce the stress condition. So reduce antioxidant and increase the free radicals will lead to the formation of glycated protein, which produced due to the hyperglycemia are responsible for the initiation of lipid peroxidation that damage the normal architecture of the cell. So GS possess strong antioxidant property, protects against the formation of lipid peroxide, and can control diabetes by obstructing oxidative stress. From the result, diabetic control group shows a significant higher levels of marker enzyme, but in GS treated group, it helps to protect the hepatocyte from damage, and also it maintains the normal level of liver enzyme. Continue with methylglyoxal is a main cause of diabetes, which the precursor of advanced glycation and product AGE. MG is highly reactive and its concentration will increase when its precursor like higher glucose increase. In major type of diabetes, the pancreatic beta cells will severely affect along with insulin secretion, meaning it's maintaining the beta cells alive is very important to keep the glucose level in a controlled manner to reduce the risk of diabetes. So histopathological analysis of pancreas clearly shows the anti-diabetic effect of GS. Due to overproduction of free radicals, insulin reducing beta cells containing islet called islet of Langerhans got damaged and steep decrease in the number of cells was observed in diabetic rats, as CS possess a good antioxidant capacities. So this helped them to protect the pancreatic islet, thereby we observe a structural improvement of cells. Next, for the conclusion, I will conclude that from the observation, it is clear that the novel herbal formulation enriched with multigrain, referred as Glymin sprinkle, can control diabetes significantly by modulating the blood glucose level, HbA1c level, liver enzyme markers, oxidative stress, enhance the glycosyllase level, and maintain the normal histology of pancreas. Thus, it can be used as a promising anti-diabetic dietary food supplement. So I think that's all from us. Thank you.